So welcome everybody back to PD2K Links Data Science Research Webinars. Today, Chan Zhu from University of Maryland, Baltimore will talk about biomedical data integration. Please, Chan, go ahead.
Okay, thank you so much for introduction. Thanks for invitation. So it's my pleasure to join this webinar to give a talk to introduce some of the efforts we have made in terms of biomedical data integration toward quality healthcare delivery. So first, I just want to introduce briefly about myself. So my name is Chan Zhu, assistant professor in health IT at University of Maryland, Baltimore County. So I have been working in this particular area about more than 10 years, including 2 postdocs I have done in Indiana University and the Mayo Clinic. So one of my research interest is to develop an integrative framework. So this framework will be able to integrate a large portion of biomedical data. So all the audience just sitting in this webinar has much better sense and idea about the big data than it's better than me. So the list of the numbers I put here is not just give you, just emphasize why in the big data area, just want to give you some sense like how diverse type of data we have and of course how large data we have. So given this a large data set from the different resource of the data, so most of the data stay in their own original format or syntax. So it's very hard to utilize all of this type of data to support our own research. So by integrate that, so one of the efforts and one of the interests I have is how we can normalize this type of the data by using standardized terminology, and how we can represent them in a meaningful way to support or efficiently to support our own research. Then we'll facilitate the data integration. So besides this data side, we're also interested to integrate some of the web surveys. We can be part of this framework. Like for example, some of these web surveys we have created, like some of the prediction models we can integrate in this. We can utilize such results to present to the end user. And also, in another part, how we can aggregate some of the computational tools. For example, like the visualization tools, I know there's a lot of fantastic visualization tool has been created in LINKS program. So that, of course, will help us to more understand or manage all this type of data. Also, we're interested to how we can help the user to use some of the intelligent tools to integrate this data by themselves. Of course, on top of that, we are interested to create some of the clinic decision support systems where you utilize all of this type of the data and the service on top of this framework. Then we will be able to achieve these 2 applications, precision medicine and drug discovery. So I will next I want to talk about some of the efforts we have made for the integrative application we have built for the drug discovery. I will talk a little bit about 3 of the systems we have built. One is called WENDI, another is a Chemogenomic Explorer and ChemoHub, and some others I'm not just including in this talk. So the WENDI is standing for Web Engine for Long Obverse Drug Information. So this project is funded by Eli Lilly company. So they just want to us to create some of the web engine can aggregate all kinds of the data, can present to their scientists, then the scientists will know the comprehensive information regarding to one of these interesting chemicals they have. So WENDI is actually based on the web service infrastructure. We just add 4 parts of this and aggregate them together. The first is like the database, which is stored in large portion of the data, and also we include some of the prediction models and some of the checks information and warp information. So this is actually is a very WENDI architecture. So we have the WENDI web interface. So the user can draw their chemicals through this molecular editor and translate into the smiles. So because WENDI takes smiles as input, so then they will send the input to the WENDI web service running in the backend. So there's 2 parts is running simultaneously. So one is for searching the data which is stored in the database. So we'll do some chemical structure similarity search or substructure search against a large portion of data. I will have the following slide will show what kind of the data we include in this database. And on the other hand, we have generate some of the prediction models, like we use the NCI cyanide model and toxicity model and some of the 3D structures model to retrieve these predictions for this particular query compound. After a while, all this information will aggregate into one XML file and send back to the user. Also, we include some of the other interactive display like the CSV and PDF for the lady scientist. So all the information has been aggregated into the XML file. And in terms of that, because we saw this huge amount of data we have already integrated for this particular chemicals. So we're thinking whether we can just translate into the RDF network, then we can run in some of the automatic reasoning to identify some of the new associations on top of that. So this is another application I will talk about later. We call chemogenomic explorer. So this is all the data we have integrated for the WENDI. So like we have the NCI tumor cyanide data, we have natural pulp scan data and drug bag data, and some mania is literature data and some of the chemogenomics data. Another one I want to emphasize is we call Chem2x2 RDF. So this is linked open drug data. So actually, we integrate a huge amount of data from the different data sources and publish as the web data. So you can see at the time of the first generation, we have over 80 million triples has been integrated into this Chem2x2 RDF data set. And there's a different type of data set concept has been used, like chemical compound, protein genetic, economics and literature, and also we have some of the systems biology data like the cake data, pathway data, and cider like the adverse events drug data. So there's a lot of data has been integrated into this one single resource we call Chem2x2 RDF. So besides that, we also have included some of the private data from the lady, like I just list the one data set we here is we integrate some jubilant journal data from the Eli Lilly. So totally we have more than 31 million chemical structures and 59 million bioactivity data points and about 19 million publications, around 500 drugs have been integrated for the WENDI's use. So this is an example to show you how what kind of the results you will be expected to retrieve from the WENDI system. So on the left-hand side is a screenshot I taken from especially based on the prediction models we generate. Also, sorry, this is a running for the one of the query compound is warfarin. So basic information about the warfarin I just post here. So on the top of that, you can see this is one of the prediction models we created based on the NCI tumor sequence, which the data we retrieve is from the NCI DTP program. So originally they have a 60 human sequence, but just because the computational cost, we just generate 40 random forest prediction models for each of these sequence. So then we will be able to have the predicted for the predicted probability of the activity of this compound of the warfarin in the individual sequence. So the different color just show the different activities predict. And under this, we have the toxicity models to predict some of the toxic effects for these particular chemicals. That's just based on the Kramer decision tree. And here I just point out here is we just adopting the top tree plugins. So to do the prediction. So both of them just based on some of the fragments has been studied for well-known toxicity. And besides prediction model, like I showed, we have integrated out of the data set to search this screenshot I take here. This here is we search for the similar compounds from the Kimball database. So you can find the similar compounds which are similar to this warfarin and the structure and similarity. And also we include the EC information and target information, also relevant literature. You will be able to find the literature information regarding these associations. So this is just a very brief introduction about the WENDI. So then I want to move to the chemogenomic explorer I just mentioned before. So like I mentioned, we have aggregated a huge amount of information regarding to these chemical compounds. So we're thinking whether we can just identify some of the novel associations from this aggregate results. Like for example, I put a flowchart is here. Like we have query compounds. We found the chemical compounds is similar to another chemical compound with PubChem compound ID. And both of them, one is active in particular AC and one is containing one of the literature. And another one we see is this query compound is predict inactive against these HER2 genes. So all of them will be rich, just point to the breast cancer has association with breast cancer. So we're wondering whether we can just establish find associations between the query and the breast cancer through this indirect links, which has already presented in the WENDI results. So this is a motivation. So we just built on our ontology to identify this relationship, to present these relationships, and a large RDF network can be expanded out starting from the query, and RDF inference engine can be applied, and results can be filtered and prioritized. So this is I will show another screen to see this fancy browser we created to although the user can auto flexible filtering based on the results. So this is a very simple structure to show you the chemogenomic sectorial ontology, the relationship. So we have a very simple structure. We have just defined 5 primary class, like the chemical compound chain, disease and bioassay, and journal articles. And also we define the relationships to make this connection from the different instance data or class data. And finally, we will be able to infer or identify the relationship between the chemical compound with genes, disease, bioassay, and the journal or literatures. So once we have created this ontology, so like I say, one of the goals to build this ontology is we can run in some automatic semantic reasoning to identify the level associations. So one of these things we just generate some of the inference rules, we'll be able to infer the relationship automatically. So I just list 3 simple rules here to just give you some of the initial idea. So like the first rule is present a very simple statement, is if the query compound is similar to another chemical compound and this chemical compound is active in particular bioassay, so then we may draw a conclusion to see this query compound might be active in this bioassay. And the rule 2 is to show if one of the diseases is associated with one particular gene and this gene is related to one compound. So then we may see the compound might be related to this disease. And similarly, for the rule 3 is the chemical compound is active in one of the bioassay, and this bioassay has been tested this particular gene, and this gene is associated with this disease. So we may see this chemical compound might be related to this disease. So this is just 3 examples to show you what kind of the rules we have defined. And this is the example to show how this instance data has been presented on top of this ontology. So this is to show one of the chemical compounds is similar to query compound we choose is methoxysteride. So also they have a chemical structure similarity is 0.929. And they have a bioassay information, they have a gene information, and the disease information. So based on these statements we have and we generate for this chemical compound, methoxysteride, then we will be able to run the semantic inference automatically against the 2 rules we have presented. I just presented in the previous slides through the rule 1 and the rule 3. Then we may just find the association between methoxysteride and the lymphoma through the intermediate layer of the bioassay with the AID number is 14 and one gene CYP1A2. So of course this just some of the recommendation or predictions we found. So further evaluation to prove this association may be recommended. So this is a fancy browser. I mentioned it before to let the user can just do some flexible filtering based on their interest. Like this is still results generated for the methoxysteride. So like for example, this screen just show we want to see more information. We retrieve for the autistic disorder related to the methoxysteride. This is social information to present like gene information, gene family info, and journal information. And also, like I mentioned, so all this application is built on top of the ontology we created. So besides we can create we can provide the fancy browser to let the user to make the selection. We also think whether we can let the user to customize their own queries, because I see this is is based on the RDF-OWL, so the query will be the SPARQL query. So we doubt most of the scientists will maybe have such knowledge regarding to the RDF-OWL SPARQL query. So we're thinking maybe we can provide them interactive web interface to let them to make their selection or customize their own queries. So then we create this SPARQL query builder again on top of this ontology. So then the user will be able to select or customize their SPARQL query. For example, this example to show is we want to find the relationship of the similar compounds for the mesosurgery with a particular gene receptor. So then we just start from the chemical compound. Then we want to find something. We want to see whether there is something literature contains such information. So the user can select stuff from the subject to select the chemical compound, and it's contained in a journal article. Then we can specify the journal article maybe has a title with this. You can put the text here. We just put 5-HT as one of the specify the genes maybe included in this journal article title. So then they can just generate the SPARQL query and submit the query, and then they will be able to find the information from the just chemogenomics explorer ontology. Then we can see this information here. So this is a brief introduction about this application. So next one, I just want to talk about, we also interesting to build some of the open platform which will be able to do some predictions, see how the strengths of the relationship between the chemicals and the genes. So we call this is a ChemoHub. So we just utilize open data, like this is the data we have already introduced in the WENDI application. And also we want to build some of the open models based on some of the RESTful services. And also we intend to make that as the open source. So this is some of the screenshot I just put here is, so which all of you, so the ChemoHub just takes the input. One is a chemical compound. You can put the CID number, PubChem compound ID number, all the smiles and drug names. And for gene, you can put the gene symbol, gene name, and UniProt ID. So then you can select the prediction models we include here. So you can just generate, you can just find some of the results to see, to assess what's the strengths of this particular two associations. Like, for example, this association, we try to find the triglyceride with this particular genes. Then we can find they have, based on this prediction model, we have very weak associations. And this is just to show you how these two nodes can be linked together and through the intermediate layers. So this is just show the network, and also for each of these nodes and edge in this network, we will be able to click on that. We will be able to find more detailed information through the Chem2by2RDF. We create some of the REST services to allow you to dip into that to see what's the detailed information from the different resources, but it's through the Chem2by2RDF. So this is all three applications I just want to introduce to you, so how we can integrate this kind of the diverse data or web service or tools together to support the drug discovery. And so next I just want to talk a little bit more about some of the other data integration efforts we made specifically for the drug repositioning by using like the medication data, pharmacogenomics data, or systems biological data. So this is one of the projects we are just working right now as, so like we know there's a lot of drug data available and a lot of efforts has been made to integrate this different integrate the drug data from the different resources. But we're thinking whether we can integrate the drug data from the drug class level, and we will be able to identify some probably we'll be able to find more just local associations between the two drugs from the different drug class label. So we are just using, like, one of the drug class data is ATC and NDFRT. So we generate these drug mappings, and we create the drug class mapping by using between these two resources. And we also integrate some of the clinical drug data from the structured product label and axonal as just a second layer. Can just add more can add more clinical drug information under each of the drug class we created. So also we, beyond the drug class or individual drug mappings, we also want to see whether we can just regroup the drug data based on their chemical structures. So we calculate the chemical structure similarity to regroup some of the drug data based on that. So then we create we call the pharmacologic class profile in our, we call the PCPO. So this is a snapshot to see how we can, and how this ontology represents that information from the different ontology, from the different resources. So this is just an example to show you how this PCPO can be used to identify some of the level drug associations. So this is the original ATC class network is presenting here. So we can see one of the drug we are interested in is Medrysone, which is only have one link to their parents, to their drug class. But after we do the integration with a different type of the data, like from the NDFRT, from the SPARQL, from the axonal, and also adding the chemical structure similarity search, we will be able to add more collections to these particular drugs. Particularly, we are interested to this association between Medrysone and hydrocortisone, and which is included into these highlight connections. So this is a drug file for Medrysone generated by the PCPO, and establish the relationship between the Medrysone and hydrocortisone. This is to show you how this ATC data and SPARQL data and axonal data along with the FRT data can make these connections and build such connection and make this inference between these two drugs. So like I see, we are also interested by using the pharmacogenomics data to support the drug repositioning, because the pharmacogenomics data give us more information regarding to the drugs, genes, SNPs, disease, and so on and so forth. So this is actually initial studies we're studying with 23 FDA-approved breast cancer drugs retrieval from the NCI, and we integrate the pharmacogenomics data from the PharmaGKB and from the drug bank and from the GWAS catalog, and also from the FDA biomarker tables. And also we also consider, like I see, we do the drug repositioning, so we still just consider the chemical structure, the drugs with similar chemical structures maybe have the similar chemical properties. So we are just also we integrate some of the cheminformatics approach to adding in this profile. Then finally, we generate the pharmacogenomics profile for 18 breast cancer drugs, and we convert that profile into our ontology, and we do the semantic inference, and we will be able to identify some of the drug candidates for the drug repositioning. So this is some of the RDF presentation for the pharmacogenomics profile we created for this 18 breast cancer drugs. Like, for example, we have a tamoxifen, which is defined as a drug, which is associated with this gene and BRCA1. And also this is associated with disease breast cancer, associated with particular pathway. And also we identify one of the another chemical, drugs, clomiphene has the similar chemical structure to the tamoxifen. And also there's some other information regarding the SNPs and other information regarding to this clomiphene. So the same as previously introduced, we want to run some automatically inference to identify some of these drug candidates. So we generated some of the rule to inference rule to facilitate this this purpose. So this is I just listed two of these very simple rules we defined to find some of the associated disease or alternative disease can be used for the tamoxifen. So like the disease can be associated, one of the another disease is associated with drugs, which is tamoxifen. All this disease is associated with other tamoxifen associated SNP or genes or pathways. And by using chemical structure, we also can simply to define inference rules like one of the diseases is associated with one of these drugs, and this drug is a structure similar to this tamoxifen. Then by using these inference rules we predefined, then we can automatically run the inference engine to retrieve this local information. So the next, probably just to talk a little bit about some of the data integration efforts we have been made for, particularly for the towards the precision medicine, especially for the pharmacogenomics data and clinic data. So, so first I want to talk about is harmonization and semantic annotation of the data dictionary from the PGN. So this is this project is under the PGN, is a pharmacogenomics research network. We are just one of the network resource under this PGN. So we the role of us is try to harmonize this data, pharmacogenomics data collected by different PGN sites and facilitate their data reuse. One of these purpose of this network resource we are working, we were working before is. So like this is one example I show here is one the data we collect from the PGN sites is, so there's two sites, they just using the different data elements to represent even this exactly same things, like for example they represent the rings and ethnicity. They are using the different variable names or descriptions, even they are using the different values to represent the same attribute compared to the standards we should just follow by the OMB. So this is a problems will be hinder the data reuse or data sharing across the different PGN sites. So we propose two approach. One is can utilize some of the standard terminologies to just normalize this type of the data. Like we can use, for example, this is one of the web interface we created for them is they can just utilize some of the standard terminology, like for example, stomacity, NOIC, and NCSers. So there's another more just terminology I have not just included in this screenshot. So then they will be able to use this different terminology to represent the different concept. Like for example, we can use stomacity and use their standard concept ID to represent the disease information, use the NOIC to represent some of the laboratory test information, and so on and so forth. So this is one effort we made to help them to harmonize this data by using the standard terminology. In the meantime, we are also considering whether we can adopt some of the data model, standardized data model to represent the data in a meaningful way. Particularly, we choose this clinical element model, so which provide a consistent architecture for represent clinical information in EHR system. And from this snapshot, you can see they represent the logic. They put the logical statements that can facilitate the consistent data representation, interpretation, and exchange. So based on this example, you can see this is a clinical element model for by using for the capture the information regarding to the blood pressure. So they have this blood pressure panel model. You can capture. They have a specific attribute called items. You can capture this systolic blood pressure and diastolic blood pressure and with these detailed values. And also they have some of the qualifier to capture the body position. So how you just take this blood pressure while you're sitting or you're just standing or whatever positions you have. So we just come up this using this standardized data model to capture this information in a more standardized way. So this is just the example to show we collect a lot of information, a huge amount of information from the PGM sites. So we want to use this clinical element model to harmonize this data in a more generalized way. So for example, I put this example here. So we have a different just a representation to show this matrix is what the age the patient has been diagnosed with particular disease. So then we will be able to harmonize these different data elements into one of the we call the single patterns like the age and disease. So then we can use capture the disease information and patient information, particularly for the age. Then we can just reuse this different clinical element model. One is the disease or disorder element model and patient demographic information. Then we will be able to present all this information in a very generalized and standardized way. So this is the effort we made, some of the effort we made by harmonized and standardized some of the pharmacogenomics data. Once this data has been integrated or just standardized or normalized, so this is another just a project just want to introduce is so how we can just made to represent and integrate time-related pharmacogenomics data with the EHR data which is stored in the EMR systems. So we'll just come up some of the temporal information and incorporate with some of the domain ontologies and which we can just transfer into our ontology and search against some of the EMR systems we created in a triple store. Then we will be able to answer some of the questions to help the physician to make the decision. So this is some of the example to show you what kind of the temporal information we have and we generate based on the data we have. Like we have the time point, time interval, and so on and so forth. And here just a list some of the examples we extract from the original data set. So once we have that, then we can transfer, we can just incorporate some of the domain ontology, especially for the temporal ontology, to represent or capture such temporal information in a meaningful way. So this is an ontology being used to represent such information. And on top of that, still we just modify the SPARQL builder we created before, as we can customize or based on the physician or whatever and user's interest. Like for example, this is want to see some of the patient information with the particular, the particular diagnosis state for the we want to find, this particular example to show the particular date of temporal information regarding to one of the patients has been diagnosed with hypertension. So then we will generate this query, then we will be able to retrieve the information against the EHR systems. So the next project I just want to introduce to you is one of the ongoing projects we are working right now. Is we call the iGenetics. So this is still based on the data integrations efforts. So the iGenetics is for the genetic test recommendation system for the individual patient. So this is actually starting from working at Mayo Clinic. So we have this the conversation with the clinicians at Mayo. So they all of us know, given the advance of the genomics technology and the biology technology, we have a lot of genetic tests available right now. But the based on the conversation we have is most of the clinicians cannot just positively incorporate with this genetic testing into their regular clinical settings because they have very insufficient knowledge about this genetics. So most of the time they have to get back to the genetic counselor to get some of the recommendation from them. Then they can just make the decision. So this maybe just have some of this diagnosis time delay and insufficient conversation. So then this is a motivation. So we want to build integrative data model and or system application can integrate the public data which regarding to the genetic test, and also combine some of the patient data extract from the EHR systems. So this is the first version we have is created for the to integrate the information for the genetic test, as we call the GTO, genetic testing ontology. So the first version we have is we integrate the genetic testing registry data from the NIH, and also the human phenotype ontology, ClinVar, and also we integrate the literature data extract from the semantic medicine. And right now we are working on this part and gene review data and also the EHR data and also the drug use data. So this is an example to show how the GTO can represent the information regarding to the genetic test. Like for example, we have the Armstrong syndrome, which has been defined as one of the genetic disease, and this have some of the clinical features associated with like obesity, and also they have associated genes, and also we have the genetic test information to show this test can be used for this particular disease and also associated with these genes. So, and also, like I said, we include some of the reference to give more scientific support to show what this association has been published in the literature. So all this is the efforts we have made and all we are still making right now by maximizing the capability of the integration to support further intelligent biomedical application can be used for the drug discovery and precision medicine. So get back to the LINKS program, we are thinking, so there's a lot of efforts we may made for the LINKS program, especially for the data representation and data integration perspective. I just list several things. Maybe you are interested in. So one thing, so we may just adopting some of the data model and or the control vocabulary to fully capture semantics of this links data, including especially for the provenance data. Like I mentioned here is a narrow publication data model. So this is a narrow publication data model as it has a very simple structure. They just include the assertion, which capture the claims you have by representing into the RDF triples, like subject, predicate, and object. And also on top of that, you can add more information and another label we call the support label to put more supportive information regarding to this association you created. And also you can put the additional label is capture the provenance data, still just regarding to this association you create. So this is a very simple example to show how this lateral publication can capture such information. So for example, we have these associations regarding to this drug is indicated for breast cancer, and we will be able to add some of the provenance data. So when this association has been created and how we create it, so and so on, you can add more if you have more information regarding that. So this is a very simple example to show you how we can utilize this information to capture such semantics. And also we can publish the links data, some of this links data in RDF4 to enable further integration with other web data, because we know we are grows the web data. We have a huge data set has been integrated into this link web data. So if we can utilize or leverage such capability from the linked open data, there will be able to retrieve more promising findings. And also, we will be able to just like the interactive visualize some of the RDF-based Linux data to help them to grab the information in more interactive way. And also like the fancy federal query will enable to further integrate the Linux data with other web data to maximize the usage of Linux data. So I will end up here, and thank you so much for your attention, and I will be happy to answer any questions you have, and or you have any just a suggestion or comments regarding the work I'm just presenting. And thank you so much.
So I would like to ask one question. I was wondering about machine learning. So you're using those RDF models, but you can also take that drug attributes and make predictions using machine learning approaches. Have you thought of doing that?
Yes, actually we do some of the machine learning approach integrates the data right now. Like we're doing some of the target modeling to try to identify some of the working with the drug profile and the gene profile created for some of the disease associations. We're running some of the prediction models right now. But like I see, I'm just not include everything here.
Are there any websites that we can go to look at the data that you organize? Is there a place where all those drug attributes, for example, can be downloaded and the RDF data sets are available for download and processing?
Some of the data set is available. I can show with you later, but some of we are still in the processing. I think we have several ontology. We have already just loaded into BioPortal. I can share the link with you after this talk.
So if there are no other questions, I would just like to thank Chan again for a great talk, and thank everybody for attending the webinar. See you next time.
Yes, thank you so much.